Good day. My name is Hayat and I'm from the University of the Witwatersrand, Johannesburg, South Africa. Thank you for the opportunity to present some of my PhD findings. My PhD is titled The Contribution of Common Genetic Variants to Breast Cancer Risk in South African Black Populations. I will be presenting some preliminary results on our genome-wide association analysis. Globally, breast cancer is the most common cancer in women and the second most common cancer in South African Black women. Breast cancer can be familial or occur sporadically in populations. About 5 to 10% of all breast cancer cases harbor rare mutations in high penetrance genes, such as BRCA1 and BRCA2. Both genetic and environmental factors contribute to the risk of breast cancer in the general population. Genome-wide association studies, or GWAS, have identified about 200 common genetic variants which contribute to breast cancer risk in populations of European origin. But little is known about the role of common genetic variants in resident African populations. The focus of this project is to identify common genetic variants which contribute to breast cancer risk in African populations by conducting a GWAS in Black South African breast cancer cases and controls. Ethical clearance was obtained for this project. My sample consists of 2,557 breast cancer female cases from the Johannesburg Cancer Study, or the JCS, and 1,074 population female controls from the Awigen Population Study and the JCS. The genotyping was all done on the H3 Africa custom SNP array, which was designed by African scientists for African populations. The array consists of 2.3 million SNPs. The JCS samples were genotyped by King's College London and the Awigen samples by Illumina in the United States. The chip data underwent quality control and imputation was carried out with the Sanger Institute pipeline using the African Genome Reference Panel. The imputed SNPs were tested for association with breast cancer using GEMMA. This model included individuals with less than 10% European ancestry and individuals with less than 5% East Asian ancestry. Population structure was accounted for using 4 principal components, and relatedness was accounted for by GEMMA, with several platforms used for data visualization. These are some of my results. This figure is a principal component analysis plot displaying IgG decomposition of breast cancer cases and matched population controls of PCs 1 and 2. European and East Asian ancestry populations are also plotted. It clearly shows that our cases and controls are well matched. This quantile-quantile plot, or QQ plot, compares the genome-wide distribution of test statistics with the expected null distribution. The genomic inflation factor lambda should be close to 1. The lambda value for this QQ plot is 0.976, which indicates good correction for population structure and genomic inflation. The Manhattan plot shows dots for the p-values for association of each SNP with breast cancer on each chromosome. A dot that is above the red line indicates association at genome-wide significance, and a dot above the blue line indicates suggestive association. A tower of dots in the same region indicates multiple SNPs supporting association at that locus. Some of the genes closest to or within these signals are indicated. The strongly associated SNPs on chromosome 2 are only present in African populations, which suggests that it is a novel breast cancer risk locus which may be specific to African populations. This is an example of a fine-scale plot known as a locus zoom plot for a top hit on chromosome 11. Multiple SNPs support the association at this locus. It is an intronic variant. Our results indicate that common variants contribute to breast cancer risk in a resident African population. Several genetic associations were detected at genome-wide significance, and at least one is likely to be a novel association. Fine-scale mapping of strongly associated loci and bioinformatic analysis of genes and SNPs in these regions is in progress, and initial analysis of known breast cancer risk loci from European populations show limited evidence of association in the South African Black population. I would like to thank everyone who has been involved in this study, without whom this would not have been possible. Thank you.